your book did a really weird thing with photosynthesis. It put the photorespiration part in chapter 5, and then it put the Calvin cycle part in chapter 6, and so I had to cobble it together, this lesson, from a couple different resources, so we'll see some different organization, but as long as you pay attention to this lesson and go through my slides for studying, you should be okay for this segment just from the slides, okay? I know the book's kind of overly disorganized and overly complex, so the lecture notes should suffice for what I expect you to understand on photosynthesis. So the last topic that we went over was glycolysis. And so that was a pathway that was conserved in every life form as a means of generating ATP from a simple carbohydrate like glucose. The rest of this semester is all going to be looking at different ways of creating energy, and we, as biochemists, we put these kinds of studies under the umbrella called metabolism. So metabolism has two parts to it: making energy and using that energy to create useful products for the life form. So we break those two into anabolism and catabolism. So looking at this chart here, we can see that catabolism is things like glycolysis, where we are generating energy in the shape of ATP, NADH, and if we happen to be a plant, also as NADPH. Whereas anabolism is using that energy of ATP, NADH, NADPH to make molecules for the rest of the life cycle, such as DNA, carbohydrates, proteins, and fats. So glycolysis was a catabolic event. Photosynthesis is a coupling of the catabolism using the sunlight to generate energy, but also anabolism using that energy to create glucose. So photosynthesis occurs in two separate phases. Colloquially, they're sometimes referred to as the light phase and the dark phase. The light phase being the phase that requires sunlight. The dark phase doesn't have to happen in the dark, but it doesn't require sunlight to occur. So in phase 1, technical word rather than the colloquialism would be photorespiration. So think about respiration, that means breathing. We animals breathe in oxygen and exhale CO2, while plants inhale CO2 and exhale oxygen. In photorespiration, we're looking at how plants break water to generate that oxygen. And they do that using sunlight. In the dark cycle, they're using that inhale of carbon dioxide and the energy generated from splitting water to fuse that carbon dioxide to another carbohydrate and incorporate that new carbon into their biomaterials. So they're like gaining matter. So in phase 1, the idea is that light energy comes in from the sun and it gets converted from light and heat energy and stored as chemical energy of a proton gradient, so a H+ gradient. That gradient of concentration is then used to drive synthesis of ATP. So that's how photorespiration gives us ATP. In doing that prior step, we use water to donate electrons to NADP+. So NADP and NAD are redox molecules. And so we typically associate their reduced state as being a high energy state. So reduced is gaining hydrogens. So NADPH would be the energetic form, and then NAD+ would be the de-energized form. So again, then, once we've generated the ATP and the NADPH using the light-dependent reactions, that accumulated ATP and NADPH is used to fuel the carbon fixation reactions. They provide the energy necessary because adding CO2 actually is a very endergonic process, requires energy input. So the ATP again and the NADPH from the light-dependent cycle is necessary to fuel the energy for the light-independent cycle or the dark cycle. So we're going to start with phase 1, start at the beginning, right? We're going to look at where photosynthesis occurs within plant cells, what the importance of chlorophyll is, the idea of how the energy is transferred through the photosystems, photosystem 1 and 2, and then the final molecule of this pathway being the ATP synthase. This diagram here links together the different reaction centers of photorespiration. So as I mentioned in this first light-dependent cycle, electrons come from water. So we're starting at what we have on the kind of y-axis here is an energy scale. So we're starting with low energy electrons from water that are given over to this reaction center called photosystem 2, and they jump up in energy from sunlight. And then that energy trickles down through a series of protein complexes to photosystem 1, where those electrons get another beam of light and jump up in energy, and that trickles down, and we finally make NADPH. This is called the Z scheme, or the Z scheme of electron transfer. Personally, I would have called it the N scheme. Because where is this Z? It's a sideways Z, right? So like N. Anyways, just know this is called the Z scheme of electron transfer because we have basically 2 bumps of energy and 2 declines of energy. So electrons get promoted to a high energy state, they lose that energy gradually, stuff happens along the way, and then they gain energy again. And then finally that drives the NADPH synthesis. Another thing that is of interest, just comparing the 2 phases, so the light phase versus the dark phase, this phase is incredibly fast. The generation of the ATP and the NADPH is incredibly fast. The dark cycle, where we're using that energy to fix carbon, is much slower. So if it helps you to synthesize your knowledge, chymotrypsin had 2 phases. Which was the fast one? The first one was the fast one, and the second one was the slow one. Same thing. First one fast, second one slow. Makes the memorization easier. Okay. So, photosynthesis only occurs in plants and photosynthetic bacteria because only those organisms have this organelle called the chloroplast. So this is like a tiny cell within the larger plant cell. And it originated from a photosynthetic bacterium called the cyanobacteria. So one of the earliest life forms on Earth were the cyanobacteria, and they were responsible for conducting photosynthesis and starting to increase the oxygen content of our atmosphere. It wasn't until we had an oxygenated atmosphere that animals and larger life forms could start breathing oxygen and exist on Earth. So they're very important little guys. But at some point during way back history of this evolution of life, a larger cell ate a cyanobacteria, and then that cyanobacteria was still alive and functioning inside that larger cell, and it was making oxygen, and it was making glucose, and the bigger cell was like, well, this is pretty sweet. I'll just keep this little guy alive, and when I divide, I'll divide him too. And so that's how our plant cells came to be. So within the chloroplast, because they're originated as bacterial cells have 2 cell membranes, not just one. They have an inner membrane and an outer membrane. Okay. And chloroplasts are special because even within that, they pancake-shaped vesicles, which are little membranes, literally pancake-shaped membranes, stacked upon each other. So we need some terms to be comfortable with going through photosynthesis. And the two terms that I want you to understand is thylakoid, that's the pancakes. Okay. Inside the thylakoid is the thylakoid space. So we have the thylakoid space is inside of one of these little granules, and then the stroma is all the aqueous water inside the chloroplast, but outside the thylakoid. So phase 1, in terms of the light-dependent reactions, they are all going to occur on the thylakoid membrane. So basically at that barrier between the thylakoid space and the stroma. So that's why I want you to know those two terms. Phase 2 also occurs within the chloroplast, but it's within the stroma, so the outer area of the thylakoids is where phase 2 is going to occur. Quick poll just to make sure we're all alive. So true or false, plants are the only organisms capable of photosynthesis. Five, four, three, two. Don't say that, Margarita, till you see if you're right or not. Okay, she was right.
That's what caused the red sea was that they couldn't do photosynthetic measurements, right? Because all of our oceans are turning red from the algae dying.
Anybody has any questions about photosynthesis, please address them to Madison.
No.
Because she's telling me things I did not know. Okay. So great, yeah, because there are also these bacteria, cyanobacteria. So all of you from the biology department, if any of you know Dr. Rowett, yes, it's Rowett, not Rawat, she studies cyanobacteria and how they actually exist at all surfaces of the earth and under surfaces of the earth. So they're very important organisms. Okay, a little bit more about that chloroplast. So we're focusing in on the little pancakes that we call the thylakoids. And so within the membrane of the thylakoid is where we see a large array of integral membrane and membrane-associated protein complexes. These protein complexes are not single proteins. So I say complexes very purposefully. So even though you may see just a green blob, that's actually composed of several proteins working together. And each of the proteins involved also contains a prosthetic group called chlorophyll. It's chlorophyll that actually contains the electrons that get energized by sunlight. So the energized electrons in the chlorophyll molecules get passed between the different proteins in these complexes, and they're going to do that going energetically downhill. So the sunlight comes in and it energizes the electrons. If you remember your Gen Chem when we were learning about electron orbitals, I see some shaking heads, it's okay. But just know the electrons are getting increased energy and they want to reduce themselves back down to their ground state. And so to do that, they have to give the energy away. And so they use that energy to do another reaction. So the reaction that happens when our electrons flow downhill in energy is that they allow protons, so hydrogen ions, to be pumped into the thylakoid space from the stroma. Okay. Eventually, when those hydrogen ions build up inside the thylakoid space, they will flow back into the stroma using the ATP synthase complex to drive ATP synthesis. So what exactly is chlorophyll? So on this slide, I have a molecule from your past, heme. Remember heme from hemoglobin, talking about how it would the iron gets pushed up by some sort of amino acid. So if we look at heme, and now you look down at this molecule of chlorophyll, you'll see they have a very similar structure. One of the key differences is the metal coordinated. So heme uses iron. We always associate iron with red blood cells and hemoglobin. Chlorophyll uses instead magnesium ions. Now this is just an example of one type of chlorophyll called chlorophyll A. There are many types. They differ kind of in what this long chain is out to the side, and some of these other functional groups like this aldehyde and this methyl group, they can be different things for the different chlorophylls, but all chlorophylls have to have this conserved ring with the 4 nitrogens holding on to the magnesium ion. And they'll also notice that the ring going around there alternates single and double bonds. Single, double, single, double, single, double. Those single and double bonds in chlorophyll are kind of similar to what you may have learned back in OChem about benzene rings. 3 double bonds, 3 single bonds. And the thing that made benzene special was because of resonance, meaning the bonds were mobile. Right? So the electrons in a resonating group are not fixed between any 2 atoms. They can kind of travel around the molecule. So if I'm looking at the chlorophyll here, the electrons from this double bond can travel to this single bond, which would bump these double bonds over to this single bond, which would bump these ones over and over. So we've got kind of a continuum of electrons moving around the chlorophyll ring. This means that the electrons are not held very tightly by their atoms, and so they're easier to energize and pass along. So it is resonance of the single and double bonds that make chlorophyll so good at passing along electrons. Okay. From molecule to molecule, yes. Yes. Intermolecular. So plants are green, right? Why are plants green? They don't absorb green light, yes. The chlorophyll actually absorbs light mostly in the blue range and some in the red range, but not in green. So if the light that the chlorophyll is pulling in is all the red and the blue, then green is still bouncing off the plant. So that's why we see plants as green, except for those weird red plants and purple plants. Let's not get into that. Wish this was plant biochemistry, but it's not. So here we can see kind of an absorption spectrum. So what we're looking at here is all of the colors of light from blue to red, and we're seeing strong absorbance of blue light from chlorophyll A and B, so it's slightly different colors of blue. And so plants will have multiple chlorophylls so they can collect multiple types of light. But you see a lot of high absorbance with this white line and this black line in blue. And then when we get to cyan and into green, it's low. And then we get kind of in yellow and red, more absorbance. So that's why plants kind of come out as looking green. So were we paying attention a second ago? So that fancy single double bond thingy was called the tetrapyrrole ring, by the way. But more importantly, What ion does it use? Three, two, one, stop. Yay! Magnesium. Cool. All right. Resonance energy transfer. It's a rather complicated phys chem kind of concept, but makes a lot of sense when we're studying photosynthesis. I told you that this light-dependent set of reactions is very fast, and I also told you that these complexes that are passing the light around, they're multi-protein subunits and lots of chlorophylls. What resonance energy transfer is the literally energy transfer between molecules that display resonance when those molecules are near each other. So what happens is we have, for example, one molecule of chlorophyll within the thylakoid membrane. And the electrons in that molecule of chlorophyll get energized by sunlight. And they go from a ground low energy state to a high energy state. And because most biological reactions are not perfect, we lose a little bit of that energy that they gained just as vibrational loss. But we still have very energized electrons that want to get to a low energy state. The whole universe is trying to get to a low energy state. So as these electrons try to travel back down to ground state, the energy that they release, if there was nothing else nearby, they'd just release it as heat or light maybe. But instead, that energy is used to drive electrons in a neighboring chlorophyll molecule up. So they're not going to go as high as the first electrons because there was that bit of loss, but they are going to increase in energy. Then they get a little bit of loss, and when they drop down, they're going to drive the energizing of another molecule of chlorophyll. So again, for this to happen, the wavelength of the light has to be properly matched. So saying the emission spectrum of the donor molecule must overlap with the absorption spectrum of the acceptor molecule. So what we're looking at is kind of like if you can think about this chlorophyll A and chlorophyll B, the peak absorbance for A is different than the peak absorbance for B, but there is some overlap where they're absorbing light. So chlorophyll A can pass energy to chlorophyll B. That's what we mean by there has to be some sort of overlap. The other qualifier is that the two chlorophylls need to be very close together. They do not actually need to touch each other. No kinetic molecular collision between the molecules are required, but they need to be real close. If we have this overlap of energy levels and they're close together, the donor directly transfers not the electron itself, but the energy of the electron. Causes the electron in the neighboring molecule to jump in energy. Fancy dipole stuff going on, not important. But because it doesn't require the collision of molecules, it can be done very rapidly because we don't have, like, if you guys think back to what perfect enzymes were, how they're limited by diffusion because you need the time for the molecules to collide. Same thing with the last step of chymotrypsin, why it's slower because you had to wait for water to get in there. If you don't have to wait for that to happen, it can happen very, very quickly. And in fact, it has to happen very quickly because otherwise the energized electron from the first molecule will just go down to its ground state and emit heat. So if you want to have the next guy getting energized, it has to happen quickly. So, way back to here, talked about how each of these blobs in this image are actually multiple proteins. So they are all organized together, bringing the molecules close together. And if I go back a few slides further to the Z-scheme, molecules within this photosystem 2, all tightly packed together, all passing that energy of their electrons. They also need to be closely packed next to these guys. So those are not freely floating all over the membrane. They're all together, maybe in a lipid raft even, pulling that out. And then up and down. So, see how good of attention you guys were paying with this question. How ready you are for this question. So in that scheme that I was showing earlier, where did the electrons start and where did they end? What was the order in the middle? Ooh, this is tricky. A, C, and D all have the same start and end. Oh, man. Five, 4, 3, 2, 1. It doesn't like you today. D. So the naming system here is what one of my old professors always says is arse backwards. We're going to go from water to photosystem 2, then plasmocyan into the photosystem 1, and then NADP is your final electron acceptor. So let's talk about the beginning of the Z-scheme, water and photosystem 2. So the electrons are initially donated from water and finally accepted by NADP. Okay? That is your start and your end. The path goes through photosystem 2, then that's Z, and then photosystem 1. So photosystem 2, again, is a collection of multiple proteins in the thylakoid membrane, and it contains a reaction center. So a specific site within that conglomeration of proteins is responsible for the energy absorption and transfer by the resonance energy transfer that we just talked about. It happens that the chlorophyll molecules that are in this reaction center have a best absorbance of light at 680 nanometers. Okay, so it's a specific color of blue that they're getting at that wavelength. So when light of 680 nanometers gets to that photosystem, the photosystem transfers a high energy electron to the next protein in the chain, which is pheophytin. So the photosystem itself, that conglomeration of proteins, loses an electron and gives it to pheophyte. Once it's done that 4 times, it's lost 4 electrons, the photosystem has become positively charged because it gave away its electrons. And so it needs to restore itself. It restores itself by stealing electrons from water. So in the water there, what we're seeing is that the 4 electrons are coming from water so that we end up with oxidized oxygen. That seems like a weird term, but it's a thing. Oxidized oxygen. So elemental oxygen has a redox state of 0, whereas the oxygen in water has a redox state of -2. So we oxidize the oxygen, and we generate 4 protons that are in the thylakoid space. So inside the pancake, we've now created 4 protons of charge. Okay? That's what happens at photosystem 2. The energized electrons then pass along this chain from pheophytin down to plastoquinone, from plastoquinone down to the cytochrome B6F complex. This is the next one I'm going to say matters. Okay. It's a complex process involving weird organic chemistry, but we're just going to simplify it down to the idea that the purpose of this B6F complex is to take the energy from photosystem 2 and use that energy to fuel a pump. Remember, charged ions can't go across membranes. We need proteins to bring those charged ions across. So this is a form of active transport where light energy is actually the energy source, driving a pump that puts another 4 protons into the thylakoid space. So we've now generated 8 hydrogen ions inside the thylakoid space, not in the stroma. While it's doing that, the electrons that have been transferred originally came from photosystem 2. Okay, those electrons were physically transferred through the chain, and they get dropped off as low energy electrons at photosystem 1. So we've had the fall from the heights of energy at photosystem 2 back down to low energy state when the electrons arrive at photosystem 1. Photosystem 1 absorbs light at a slightly different wavelength, so 700 nanometers, so a little lower in energy. Remember, wavelength is inversely correlated with energy. Shorter waves are higher energy, like UV. It's like 280 nanometers, high energy wavelength. So a lower energy wavelength absorbs at the photosystem 1. So now we have re-energized our electrons. We're going to use that resonance energy transfer again to move these electrons through another chain of various iron-sulfur proteins through ferredoxin, and those are finally transferred to an enzyme, ferredoxin NADP oxidoreductase. Oxidoreductases, we can qualify them because they always use either NAD or FAD or NADP in this case. So this enzyme catalyzes a reaction where NADP plus is reduced to NADPH. And that's going to use up 1 proton, but that proton is coming from the stroma. So we still have that collection of extra protons in the thylakoid space. So it's going to take a proton away from the stroma, and it's also going to use up 2 electrons for every 1 NADP plus converted to NADPH. So if we think back to earlier in this cycle, it took 4 photons of light to move 4 electrons out of photosystem 2, and then those were restored by 1 water. Okay. Now we have 2 electrons needed to drive NADPH synthesis. So for each 1 water, we're getting 2 NADPHs. Okay. So that's the first energy payback from photosynthesis or photorespiration. Then photosystem 1, it got its electrons back, the ones that it passed down the chain, from, as I mentioned earlier, plastocyanin. So if we look at our accounting, shoot, I misspoke earlier, it's 2 waters, right, to get the 4 photons, not 1 water. So 2 waters and 4 photons of high energy are used to make 4 protons in the thylakoid space. We generate 1 molecule of oxygen gas and 4 high energy electrons. Those 4 high energy electrons are used to drive another 4 protons into the thylakoid space, and then they go down to low energy electrons. Then at photosystem 1, those low energy electrons plus 4 more photons, they get energized. They take 2 NADPs, 2 protons from the stroma, and that creates 2 NADPH. So your net reaction is 2 water molecules and 8 photons of light are used to pump effectively 8 protons into the thylakoid space, generating 1 molecule of oxygen and 2 molecules of NADPH. Just making sure I mentioned everything I wanted to do that. Oh, going back to this slide, there was something I didn't know earlier. So again, cytochrome B6F. As the electrons are coming down in energy, this guy is pumping 4 protons into the thylakoid space. Right? Protons have what charge? Positive. What we're trying to do here, what the cell is trying to do here, is create a proton gradient, not a charge gradient. Okay? It's trying to create a pH difference. So when we pump those protons into the thylakoid space, at the same time, magnesium ions are being allowed to float out into the stroma as counter ions to counter that charge difference. Okay, so finally, we get to the real money step of photorespiration, and that is the generation of ATP. So back to our accounting, we use those 8 photons of light to get to this point where we've generated 1 O2 and 2 NADPH, and we have 8 protons in the thylakoid space. So 8 photons of light have effectively been used to move 8 protons into the thylakoid space. This actually is enough to create a pH difference between inside the pancake and outside the pancake. So inside, you're pushing protons in. So where's the pH lower, the inside or the outside? The inside, right? So it generates a difference of over a whole pH unit. 1 to 1.5 pH units difference. That chemical gradient can be harvested as energy, just like we learned about with axons, creating the sodium-potassium gradient. And then if we want to fire the axon, we just open the gates and allow that chemical energy to drive equilibrium. Same kind of idea here. We have another set of proteins called the ATP synthase complex. And it's this wonderful mushroomy-like thing here. And so it's composed of the F0 version, which is the membrane channel portion, F0 portion, and the F1 portion. Protons flow through this F0 portion, and it's so cool. What happens? You're adding a proton as it goes through the channel. It protonates an aspartate. What does aspartate have of interest? What kind of amino acid is aspartate? Acidic, right? So it normally at neutral pH has a COO minus. So when the protons flow through, it gets to be protonated for a second, COOH. That makes it less polar, and it actually rotates. So each of these tubes here are like a ball bearing that can rotate as the protons go through. It's kind of like if you think about one of those like all the logs, and you roll the block along the logs. That's essentially what's going on in these membranes. Rolling of these F0 portions causes this part to spin, and that mechanical energy provides the energy to take ADP and PI, fuse those back together to generate ATP. So the payback is actually from those 2 waters and those 8 photons, we get 3 ATP molecules. So you need to know what your terms are. Which one was the stroma? Is that inside the pancake or outside the pancake? 5, 4, 3, 2, 1. Right now we have equal votes for B and C. C. The inside gets more acidic. That means the outside is getting more alkaline, basic. So I think carbon fixation is going to eat a little bit into our review that I have planned for Wednesday. So I'll try to do what I can with it, but I don't want to overrush it. It's not as long as the first half anyway. So we're moving on to phase 2. We've got our payout of ATP and NADPH from photorespiration. So now we're looking at the dark cycle, which is carbon fixation, the actual using of CO2 to make glucose. So we're going to use the energy from the first step to drive this next cycle, which is named after some guy with the last name Calvin. So we call it the Calvin cycle. So within this cycle, it actually is a cyclical process where we have CO2 coming in, we do a bunch of reactions with it, and then we regenerate the starting material so CO2 can come in again. So there's 3 phases to carbon fixation. The actual carbon fixation part, where CO2 is physically covalently bound into the system. Then the reduction that is used to generate glyceraldehyde 3-phosphate. And then finally, regeneration of this molecule here called ribulose 1,5-bisphosphate, which is the molecule that actually accepts the CO2. And the net reaction that we're going to have is we'll take 3 CO2 molecules and 9 ATPs, 6 NADPHs. So very costly reaction to make half of a glucose. So if you want to actually bring that out to whole numbers, it's taking 18 ATPs and 12 NADPHs to make 1 glucose. Do you guys remember from glycolysis how much ATP you get from 1 glucose? 2. That's a ripoff. All right. Luckily, plants have all that sunlight giving them the energy. So the first step of the Calvin cycle is catalyzed by the world's most interesting, most abundant most abundant enzyme, I should say, called rubisco for short. The full name is ribulose. So that sounds like a carbohydrate, yay? Ribulose 1,5-bisphosphate. So that's its substrate. carboxylase, oxygenase. We're going to be focusing on the carboxylase factor, not too much about the oxygenase factor. So rubisco is the name of the enzyme. And one of the reasons it's the most abundant enzyme in the world is, one, because there's like a ton of plants on Earth, but also it's kind of a crappy enzyme. Its Km is about the same as the concentration of CO2 in the plant's tissues. So that means, at best, it's running at half speed. Right? Km is the concentration that will give you half of Vmax. The other thing is, its Kcat, its turnover number, is that it can add 1 CO2 to this ribulose 1,5-bisphosphate every 3 to 4, it can do 3 or 4 per second. Now that might seem fast to you, but if you think about all the other enzymes we were looking at, their turnover numbers were more like 1,000 per second. So this enzyme is pretty dang terrible. So the plants need a lot of it if they're going to get the use from it. So within plant cells, it's actually a fairly concentrated enzyme because they need so much of it in order to carry out carbon fixation. Getting back to enzymology, the type of enzyme that it would be is a lyase. Again, this is one of the ones where we can understand it, where it gets its type more from its reverse reaction than the forward reaction. What we're actually doing is taking two molecules and putting them together. And we normally think of lyases as what happens when you break one molecule into two pieces. But nevertheless, this is what happens. It takes this ribulose 1,5-bisphosphate and it adds the carbon dioxide as kind of a carboxylate, like carboxylic acid group, onto one end. So this is carbon 2 of the ribulose 1,5-bisphosphate. Adds it there. This becomes an extremely unstable compound. We learned about how fructose 1,6-bisphosphate was unstable because I was telling you like the phosphates are fighting each other. So this is a similar compound. It already has two phosphates which are negative charges fighting each other. And you go and add another negative charge in the middle. Extremely unstable. So with a little water around, bam, it breaks. And it breaks in half to create 2, 3 carbon sugars. It's a lot like glycolysis. We took a 5-carbon sugar, which was this ribulose guy. He only has 5 carbons. Added a carbon to it, and then that broke. And we have 2 3-carbon products. These products are called 3-phosphoglycerate. You can also note the subscript on my slide. May be hard for some of you to see. It says that this enzyme is activated by high pH and high magnesium concentration. This is why I went back a moment ago in my slides to tell you that the cytochrome B6F complex, while moving protons into the thylakoid space, pumps magnesium into the stroma. These reactions are all taking place in the stroma. So if we have our light reaction going on, that's causing protons to go into the thylakoid space, making the thylakoid acidic, but as we saw in the clicker question, making the stroma basic, high pH. It's also, as those protons are going into the thylakoid space, it's getting magnesium into the stroma, high concentration of magnesium. So again, even though we talk about this second phase as being the dark cycle, it works best when it's happening at the same time as the light cycle, because the light cycle is creating these conditions that assist with Rubisco's activity. That is my last minute, so we'll have to do the rest of the slides when I see you again on Wednesday, and then we'll do the review for the midterm, which is on Friday? Yes, thank you. Friday, midterm.